How can we engineer bacteria to remove carbon from the atmosphere? Engineering bacteria to remove carbon from the atmosphere is a promising approach to mitigate climate change. This involves genetically modifying bacteria to enhance their natural ability to capture and store carbon. Here's an outline of the key strategies and steps involved. 1. Selecting suitable bacteria. Cyanobacteria and photosynthetic bacteria. These bacteria naturally use carbon dioxide (CO2) during photosynthesis. Cyanobacteria, for example, convert CO2 into organic compounds using sunlight. Non-photosynthetic bacteria. Some bacteria can be engineered to consume CO2 and convert it into biomass or useful chemicals without relying on sunlight. Examples include Ralstonia eutropha and Methanotrophs 2. Enhancing carbon fixation pathways. Overexpressing key enzymes. Increase the expression of enzymes involved in the Calvin cycle, e.g., ribulose 1,5-bisphosphate carboxylase oxygenase, or rubus-CO, to boost CO2 fixation. Introducing synthetic pathways. Incorporate synthetic or alternative carbon fixation pathways, such as the reductive acid-LCOA pathway, to enhance CO2 conversion efficiency. 3. Improving carbon storage capabilities. Engineering carbon storage mechanisms. Modify bacteria to enhance the production of biopolymers like polyhydroxyalkanoates, PHOs, that store carbon. Enhancing biomass production. Alter metabolic pathways to direct more carbon into biomass, increasing the overall carbon sequestration capacity. 4. Optimizing environmental tolerance. Improving environmental resilience. Engineer bacteria to tolerate various environmental stresses, e.g., temperature, pH, to ensure they can thrive in diverse conditions. Nutrient utilization. Modify bacteria to utilize available nutrients more efficiently, reducing the need for external inputs. 5. Deployment strategies. Bioaugmentation. Introduce engineered bacteria into environments where CO2 levels are high, e.g., industrial emissions, agricultural soils. Bioreactors. Develop bioreactors that house engineered bacteria, allowing for controlled and efficient CO2 capture and conversion. Symbiotic systems. Integrate engineered bacteria with plants to enhance overall carbon capture in agricultural settings. 6. Monitoring and regulation. Tracking carbon sequestration. Implement systems to monitor the efficiency and stability of carbon capture by engineered bacteria. Regulatory compliance. Ensure that the use of genetically modified organisms, GMOs, complies with environmental and safety regulations. Potential challenges and considerations. Horizontal gene transfer. Prevent engineered genes from spreading to non-target organisms. Ecological impact. Assess and mitigate any potential negative impacts on natural ecosystems. Public acceptance. Engage with the public and stakeholders to address concerns about the use of GMOs for carbon sequestration. Example projects and research. Several research projects and initiatives are already exploring these strategies. For instance, researchers at the University of California, Berkeley have been working on engineering cyanobacteria to enhance their carbon capture efficiency. Additionally, startups like LanzaTech are using genetically modified bacteria to convert industrial CO2 emissions into useful chemicals. Conclusion. Engineering bacteria to remove carbon from the atmosphere involves a combination of genetic modification, optimization of metabolic pathways, and strategic deployment. While challenges remain, advances in synthetic biology and biotechnology hold significant promise for developing effective bacterial systems for carbon sequestration. Engineering bacteria to remove carbon from the atmosphere is a promising approach to mitigate climate change. This involves genetically modifying bacteria to enhance their natural ability to capture and store carbon. This introductory statement highlights the potential of using bacteria to combat climate change by leveraging genetic engineering techniques to improve their ability to absorb and sequester carbon dioxide (CO2) from the atmosphere. 1. Selecting suitable bacteria. Cyanobacteria and photosynthetic bacteria. Natural use of CO2. Cyanobacteria and other photosynthetic bacteria naturally absorb CO2 during photosynthesis. converting it into organic compounds using sunlight. Example, cyanobacteria are particularly efficient at converting CO2 into organic matter, making them ideal candidates for genetic engineering to further enhance this process. Non-photosynthetic bacteria, CO2 consumption without sunlight. These bacteria can be engineered to absorb and convert CO2 into biomass or other useful chemicals without the need for sunlight. Examples, Ralstonia eutropha, known for its ability to produce bioplastics from CO2. Methanotrophs, bacteria that consume methane, CH4, which can be derived from CO2, and converted into biomass too. Enhancing carbon fixation pathways overexpressing key enzymes. Calvin cycle. This is the primary pathway used by photosynthetic organisms to fix carbon. By increasing the expression of key enzymes, such as ribose ribulose 1,5-bisphosphate carboxylase oxygenase, the efficiency of CO2 fixation can be significantly improved. Introducing synthetic pathways, alternative carbon fixation. Incorporating synthetic or alternative carbon fixation pathways, such as the reductive acid-LCOA pathway, can enhance the efficiency of CO2 conversion. This pathway is used by some anaerobic bacteria and is highly efficient in converting CO2 into organic compounds. 3. Improving carbon storage capabilities. Engineering carbon storage mechanisms. Biopolymers production. Modify bacteria to enhance the production of carbon-storing biopolymers like polyhydroxyalkanoates, PHOs, which are used for long-term carbon storage. Enhancing biomass production. Metabolic pathways. Alter metabolic pathways to divert more carbon into biomass, the physical body of the bacteria, thus increasing the overall capacity of carbon sequestration. 4. Optimizing environmental tolerance. Improving environmental resilience, stress tolerance. Engineer bacteria to withstand various environmental stresses, such as extreme temperatures or pH levels, ensuring they can survive and function in diverse conditions. Nutrient utilization. Efficient nutrient use. Modify bacteria to utilize available nutrients more efficiently, which reduces the need for additional inputs and makes the process more sustainable. 5. Deployment strategies. Bioaugmentation. High CO2 environments. Introduce engineered bacteria into environments with high CO2 concentrations, such as industrial emissions or agricultural soils, where they can effectively capture and convert CO2. Bioreactors. Controlled environments. Develop bioreactors that house these engineered bacteria, providing a controlled environment for efficient CO2 capture and conversion. Symbiotic systems. Integration with plants. Combine engineered bacteria with plants to enhance overall carbon capture, leveraging the natural symbiosis between plants and bacteria. 6. Monitoring and regulation. Tracking carbon sequestration. Monitoring systems. Implement systems to monitor how effectively the bacteria are capturing and storing carbon, ensuring the process is efficient and stable over time. Regulatory compliance. Safety regulations. Ensure that the deployment of genetically modified organisms, GMOs, adheres to environmental and safety regulations to prevent unintended consequences. Potential challenges and considerations. Horizontal gene transfer. Gene spread prevention. Develop strategies to prevent engineered genes from transferring to non-target organisms, which could have unintended ecological consequences. Ecological impact. Impact assessment. Assess and mitigate any potential negative impacts on natural ecosystems to ensure the approach is environmentally sustainable. Public acceptance. Engagement and education. Engage with the public and stakeholders to address concerns and gain acceptance for the use of GMOs in carbon sequestration efforts. Example projects and research initiatives. University of California, Berkeley, researchers are working on enhancing the carbon capture efficiency of cyanobacteria. Startups like LanzaTech, using genetically modified bacteria to convert industrial CO2 emissions into useful chemicals, demonstrating the practical applications of this technology. Conclusion: engineering bacteria to remove carbon from the atmosphere involves a combination of genetic modification, optimization of metabolic pathways, and strategic deployment. While challenges remain, advances in synthetic biology and biotechnology hold significant promise for developing effective bacterial systems for carbon sequestration. This final summary emphasizes that although there are challenges, the advances in synthetic biology and biotechnology are paving the way for effective solutions to use bacteria for carbon sequestration, offering a promising tool to help mitigate climate change. What products can be made from these bacteria? Engineering bacteria to capture and convert carbon dioxide (CO2) can lead to the production of a variety of valuable products. Here are some key products that can be derived from these bacteria. 1. Biofuels: ethanol and butanol. These alcohols can be used as renewable biofuels, reducing reliance on fossil fuels. Bacteria can be engineered to convert CO2 into these alcohols through fermentation processes. Biodiesel: certain bacteria can be modified to produce fatty acids, which can then be converted into biodiesel. 2. Bioplastics: polyhydroxyalkanoates, FOS. FOS are biodegradable plastics produced by bacteria as a means of storing carbon. These bioplastics can be used in packaging, agriculture, and medical applications. 3. Chemicals and industrial feedstocks: acetone and isopropanol. Bacteria can be engineered to produce these solvents, which are widely used in industrial processes and manufacturing. Lactic acid: lactic acid is used in food, pharmaceuticals, and is a monomer for producing polylactic acid, PLA, a biodegradable plastic. Succinic acid: used as a building block for producing various chemicals, including solvents, food additives, and biodegradable plastics. Acetic acid: used in the production of vinegar, as a chemical reagent, and in the manufacture of various synthetic materials. 4. Biomaterials: cellulose and hemicellulose. Some bacteria can produce cellulose and hemicellulose, which can be used in textiles, paper, and as food additives. Proteins and enzymes: engineered bacteria can produce specific proteins and enzymes for use in food processing, pharmaceuticals, and industrial catalysis. 5. Food and feed: single-cell protein. Bacteria can be engineered to produce high-protein biomass that can be used as animal feed or as a protein supplement in human diets. Nutraceuticals: bacteria can be engineered to produce vitamins, antioxidants, and other beneficial compounds that can be used in food and dietary supplements. 6. Pharmaceuticals: antibiotics and antimicrobials. Engineered bacteria can produce antibiotics and antimicrobial compounds, which are essential for medical treatments. Hormones and biologics: bacteria can be used to produce therapeutic proteins, hormones, like insulin, and other biologics used in medicine. 7. Specialty chemicals: biopolymers. Polymers produced by bacteria can be used in various applications, including medical devices, drug delivery systems, and tissue engineering. Pigments and dyes: certain bacteria can be engineered to produce natural pigments and dyes for use in textiles, cosmetics, and food coloring. 8. Renewable energy: methane. Methanotrophs can convert CO2 into methane, which can be used as a renewable natural gas. Hydrogen: some bacteria can be engineered to produce hydrogen gas, which is a clean fuel for energy applications. By leveraging the natural metabolic pathways of bacteria and enhancing them through genetic engineering, it is possible to convert CO2 into a wide range of useful products. This not only helps in reducing atmospheric CO2 levels, but also provides sustainable alternatives to many traditional industrial processes and products.